Hello and welcome to section 9.3. We have been talking a lot about meiosis and about variation in sexual reproduction and how these processes occur. In this section, we're going to look at something called karyotypes. And karyotypes are helpful in helping us identify chromosomal abnormalities. Whoa, excuse me, that wasn't supposed to happen. This is a picture of a karyotype. If we look at a cell and we look at all the different chromosomes, we can organize them into patterns to help us identify different abnormalities. So in the 50s or 60s, there was a conference in Denver, thus the name the Denver System of Chromosome Classification, where they agreed on a way to write karyotypes, to lay them out, and to classify them. And this was sort of the idea they came up with based on the centromere location, the symmetry, and the size of the different chromosomes. So you can see right here, this is often called Group A, where they will pair the chromosomes. All the chromosomes get paired because remember they're homologous pairs for each one. We have the pairs here for 1, 2, and 3. They get just a little shorter as you go down, but the centromeres tend to be right in the middle. These are a little off center. The centromeres a little bit more to one side in Group B. Group C, the centromeres are a little bit off, but they just gradually decrease in size. Group D chromosomes are very much off-center. The centrosome is, or the centromere is definitely off of center by quite a bit. Group E, the chromosomes get a little bit shorter. The centromere is more in the center, and Group F even shorter still. Here's Group G. So far, that's up to 22 different chromosome pairs, and those are all autosomal. We have right here our sex chromosomes. This happens to be a karyotype of a male. If you look at that, it has an X and a Y. So it's a male. G-banding or staining identification techniques are used as well. They match the Denver system very closely. And basically what happens is the stains will stain the different genes to different degrees, and you get these little bands on the different genes. And that's another way to be able to identify them. It's a little bit more difficult to tell exactly where the centromeres are on these, and some of them are bent a little bit funny, just simply the stress of being squashed in this procedure and in this stain. Sometimes they're not straight, but they still have the same banding pattern as their homologous pair. Here, again, this is just a G-banding diagram. It's another way of representing the karyotype, kind of a cartoon representation where the actual photograph is taken and then it's redrawn in this sort of cartoon style. to show the different banding patterns and the different genes that are represented in each of the chromosomes. So we can, once again, if we can catch that cell in the act of dividing, we can get a squash of all the karyotypes and take a nice picture of that, blow it up, cut all of the individual chromosomes out, pair up the homologous pairs, lay them out in this nice karyotype, and it's really easy to see exactly what we're looking at for the individual. This individual, I have a few corrections. The templates were wrong. This is a normal female because there's two X's right here. The 2N number is 46, the diploid number of cells. 46 cells. We have or 46 chromosomes, excuse me, not cells. We have 22 times 2 would be 44 plus 2 more with the sex chromosomes. That gives us 46. This is a karyotype of a normal male. There's one X and one Y. So again, it's a diploid number is 46 and it's an XY, a male. In class, you are going to be asked to create karyotypes out of great big chromosome models that look a lot like these. They'll be magnets, and you can arrange them in the Denver identification style, and using the G banding. The little circle represents the centromere, and you can then determine characteristics about the individual. Sometimes you can find very unusual patterns that show up. Right here is an unusual pattern for a karyotype. This is a boy, an XY. This boy, however, if you'll notice, look at the 21st chromosome. There are 3 of them. This person, this individual, has Down syndrome. Down syndrome oftentimes is called trisomy 21 because the 21st chromosome has trisomy, there are 3 of them, 3 chromosomes. So this individual has actually not 46 chromosomes, but 47. How does this happen? How do we get someone that has an extra chromosome like that? Well, the technical name for it is non-disjunction, right here. And when we talk about non-disjunction, what it means is that the chromosomes do not properly separate during meiosis. That can happen during meiosis 1, or it can happen during meiosis 2. Here, when we have meiosis 1, we have those tetrads that don't properly separate. They go off to one side together. And you can see then, if we go down to the normal meiosis and then follow that pattern all the way through to the end where we get the 4 different gametes, you get 2 of the gametes having trisomy, having an extra chromosome in them, and 2 gametes that have 1 fewer chromosome in them. By the way, if these end up fertilizing another gamete, that zygote will not survive. This is not survivable. This is another scenario where you get the non-disjunction happening in meiosis 2. So the chromosomes are not properly separating. You get the sister chromatids that do not separate. It's called non-disjunction. And you get, again, you get a gamete that has 3 chromosomes instead of the appropriate 2. And you get one that has 1 less. And these 2 then would be normal. What happens is if you get an egg cell that has that extra chromosome in it, for example, or vice versa, it could be the sperm cell, but then it carries out fertilization with the opposite gamete, you get a zygote that has an extra chromosome. And that Down syndrome is a genetic disorder that is caused by non-disjunction. So most of the time, non-disjunction disorders are absolutely not survivable. Just rarely are they ever. Down syndrome happens to be one of these non-disjunction disorders that is survivable in humans. It's absolutely the most common one. There are a few others that happen to be survivable. For the most part, however, if an individual has a non-disjunction disorder, they will not survive very, very, very early in the pregnancy. That zygote will, it's fatal. So the other disease besides the Down syndrome here, we have Edwards syndrome is the second most common non-disjunction disorder. It's called trisomy 18 because there is an extra 18th number chromosome. It is not that survivable, however. The majority of individuals that have this do die before birth, but those that are born usually only live a few years. 80% of them are female. They have a lot of health issues with kidney, heart problems, and so this is, although it's quote-unquote survivable, not for very long. These are both autosomal non-disjunction disorders because they deal with the body chromosomes. There are also several sex-linked chromosomal non-disjunction disorders, and these are interesting. Turner syndrome would be one of those. It would be where there is only a single X chromosome. It's called monosomy X. That's a female, obviously, because there's no Y. And Turner syndrome, girls and women that have Turner syndrome, I mean, it's very, very survivable. It's not a severe, not like some of the autosomal non-disjunction disorders. However, they do have a few extra health complications sometimes some cognitive difficulties. Typically, they would not have a menstrual cycle. They would not be fertile. A few of the characteristics that might be common with this syndrome would be a shorter stature, broad chest, neck webbing. Then oftentimes they would have surgery for that, lower set ears. It may or may not be very pronounced, but those are some of the typical symptoms for that. Klinefelter syndrome, it is one where there is an extra X chromosome. So it's a boy, right? Klinefelter individuals that have Klinefelter syndrome are male because they have the Y, but they have an extra X in there. This also is about 1 in 500 to 1 in 1,000 live male births will have Klinefelter syndrome. The only reliable way to know for sure if someone has Klinefelter syndrome is through their karyotypes. You would need to do a blood test and look at their actual karyotype. You can't always tell. It's not always obvious. A lot of times the symptoms don't really appear until after the onset of puberty. They might include sterility, a little bit taller than the average male, maybe a little more breast tissue, maybe a little less muscle control or coordination, a little lower testosterone levels. Sometimes they're more likely to have other health problems as well. But obviously they would have an extra chromosome, so they would have 47 instead of 46 chromosomes in their cells. The next one here is super male. If an individual is born with an extra Y, so if we had a non-disjunction disorder of the Y chromosome and a man gets an extra Y, he's still a man. It's kind of a super male is what it's called. Most of the people that have this don't even know. There's a normal phenotype. There aren't really anything glaring obvious characteristics. Sometimes individuals might have a little more of an issue with learning difficulties, but most of the people don't even know, and you wouldn't know unless you had a blood test. The super female, I realize there's a typo there, triple X syndrome, where the female gets that extra X chromosome. So she also has 47 instead of the 46 chromosomes. Again, you wouldn't know unless you had a blood test and you were able to pinpoint that. You probably would never know that if you were a triple X female or not. Sometimes they are, sometimes they have learning difficulties. It's about 1 in 1,000 girls will be born with the extra X chromosomes. Sometimes they're a little taller, but they're fertile. Everything else is really very normal about someone with an extra X chromosome. So if you want some extra learning, you want to do some advanced proficiency, ask some questions about some of these things. It's interesting. See if you can do some research and ask your own questions and find some answers and come and let me know. So we'll see you in class to do the karyotyping activity.